But why don't we move on to the next speaker, and that is Nico Tromas, and Nico is also has received his PhD in Spain and is now in Canada at McGill University, and he's doing his work with Dr. Jesse Shapiro, so we'll let him explain what he's doing.
Thanks, Lisa.
Yes.
I will share already the screen. All right. So can you see the presentation?
Yes.
All right, perfect. So hi, everyone. It's a pleasure to present this work at this symposium. And I would like to first, thank you, Lisa, and also all the team organizing these events. And today I will present a work that we published recently, where we describe the composition and the association of heterotrophic bacteria that are physically attached to the mucilage of cyanobacteria, and that we define here as its microbiome. So a few years ago, I was in the lab. I was starting working with cyano at this time, and with my friend and collaborator, Alessandra Gianni. And she was telling me that when cyanobacteria are maintained in during a long time with different passages, they might lose their capacity to form colonies. So I asked her why, and one of her hypotheses was that potentially during passages, the culture lost heterotrophic bacteria that are initially present with the colony. So I was really surprised, because first, it means that many cyanobacterial culture might not be really actinic. And secondly, that when you start a culture from a single colony, it means that heterotrophic bacteria are still attached to the cyanobacterial colony. So can cyanobacteria have their own microbiome? And to answer to that question, so we did this study where we focused on the bloom-forming Cyanobacter Microcystis. And this work was led by Olga Perez here and myself, where we both worked with Jesse Shapiro at Université de Montréal. So the term phylosymbiosis here refers to when a pattern when microbiome composition mirrors the cyanobacterial host phylogeny. So just a few pictures to show what is a microcystis bloom. Here in the middle is, for example, a picture that we took during the summer of 2016 at Lake Champlain, when I was sampling with my colleagues and with Olga. And the last pictures is of a picture from one of our microcystis culture, where you can see heterotrophic bacteria that are with long stalk that are attached to the to the microcystis cells here. So basically, to characterize the microbiome of microcystis colony, we developed a novel approach to isolate, extract DNA, and sequence many different single microcystis colony. So in total, we sequenced around 109 single colonies from Lake Champlain in Canada and from Popular Reservoir in Brazil. We also sequenced filtered water with a shotgun metagenomic approach. So the first thing that we did was basically what is the composition of microcystic microbiome. So that's the first thing that we look for. And for that, we manually created several metagenome assembled genomes or MAGs that we recovered from each colonies. And we found a total of 18 microcystis genotypes, so 14 in Lake Champlain and 4 in Brazil at 99.9% of NI. Attached to the colony, we also found 72 different heterotrophic bacteria with an average 6 associated bacteria per colony. We didn't find any core microbiome, but we do find some highly prevalent taxa, like Rhozomonas, Rhodobacter, or Phenylobacterium. Sorry, take a bit of time. Right. So how the next question was how we can, let's say, classify or what is nature of the different association with Microcystis. So is it random or not, basically? So the first thing is we use the information from the environmental metagenomes, and we evaluated the co-occurrence between Microcystis and each of the most prevalent members of the microbiome at genus and species levels. And when we compared the results, so we compared the results with Microcystis and with the second most dominant cyanobacteria, which is the Dolichospermum for Lake Champlain. And when we compared the co-occurrence score, we found that basically the most prevalent taxa from the microbiome members were significantly co-occurrence with Microcystis, but not with the Dolichospermum. So that means that the association is non-random. Then we look at which factors could explain the microbiome composition. So one of the first questions is that we ask is if Microcystis genotypes, different genotypes could have a different Microcystis microbiome. So to test this, we first evaluate what could be the main factor explaining microbiome variance. So we include as well different environmental variables like pH, temperature, and also biotic factors. And we found that Microcystis genotypes explain much more the variation in microbiome community structure. We then assess if there is any Phylosymbiosis. So basically, if the microbiome composition is highly associated with the host's phylogeny. So we did a mental test and a procrust analysis. And we compared basically the Microcystis phylogeny distance matrix and the microbiome Brachyartis dissimilarity matrix. And we did find that variation in the microbiome composition is correlated with the host's phylogeny. Then, so we found that Phylosymbiosis signal, and we then wanted to go a bit deeper and to analyze potential evolutionary association. So Phylosymbiosis can arise from vertical inheritance of microbiome, but also from horizontal acquisition of microbiome at each generation. So we test here for the similarity between the phylogenetic tree between Microcystis phylogeny and each of the most prevalent taxa. And we did find a signal of co-speciation between Microcystis and only two members of the microbiome members, which are Rhodobacter and Rhozomonas. We also looked to other evidence of evolutionary association, and we analyzed the AGT events between Microcystis and its microbiome using different approaches like MetaChip, and we did find like 1,900 genes that were involved in AGT, enriched especially in secondary metabolites, biosynthesis, replication, or defense mechanism. Interestingly, we find very, very few AGT between Microcystis and the two taxa that previously I mentioned, as we found a signal of co-speciation. So then we just ask what could be the function of that microbiome for Microcystis. So for that, we functionally annotated Microcystis and microbiome genomes and confirm the presence of some metabolic pathway with a different combination like genome scale metabolic models that use Metastic for pathway interaction. Here you can see a PCA based on present absence of cake genes. And what we found is basically that taxa that belong to the same phylum are in general functionally redundant, which is the case as you can see between Microcystis and Pseudoanabena. We also found that Rhodobacter and Rhozomonas contain carotenoids biosynthesis pathway that are different than those we found from Microcystis, meaning that potentially the Microcystis microbiome could be involved in production of new or different carotenoids that would confer more of photo protection. Finally, we test if there is any potential interaction, metabolic interaction between Microcystis and its microbiome. So as you can imagine, we have no idea about the optimal culture condition as the colony were isolated from lake without any culture step. So this is why this kind of metabolic pathway interaction database like MetaCyc or CyanoCyc is very valuable for such analysis. So we reconstructed the genome scale models with a different approach like GAPSEC or CARVEMI, and some of them use MetaCyc database. And we use Metana that look basically that identify the different potential exchange of metabolite in a community. And we found that Microcystis was the major recipient, not a donor of metabolite, especially for some amino acids and nitrogen species from the microbiome. And we also find that Rhodomonas was basically the taxa from the microbiome that has a higher metabolic interaction potential score. All right, so just to summarize what we found in this study, there are heterotrophic bacteria that are attached to Microcystis mucilage that we define as Microcystis microbiome. We characterize this small community and found that on average, there are 6 taxa per colony and no core microbiome. But we do find highly prevalent taxa like Rhodobacter, Rhodomonas phenylobacterium. We also found a significant signal of Phylosymbiosis. Basically, the microbiome position is associated with host phylogeny. And we also find some microbiome taxa that have co-evolved with Microcystis, so Rhodomonas and Rhodobacters. And these 2 taxa showed a very low number of EGT compared to the other member of the microbiome. And finally, we also found that there are potentially metabolite exchange between Microcystis and its microbiome, mostly composed of amino acids and nitrate. I would like to thank the different members of Shapiro Lab, especially Olga and Jessie. Thanks also to my collaborator at CNRC Canada that helped for the sampling. And I also would like to finish on that side project that I'm doing with friends and colleagues. It's a children's book on 2 little cyanobacteria that we freely distribute in schools to help the prevention and information on cyanobacteria. Thanks a lot for your attention.
Thank you so much, Nico. That was great. And I wish I could grow Prochlorococcus to as dense cultures as we see with Microcystis. But also it points to all the amazing new research that's coming out on the interactions between the microbial communities with the cyanobacteria. So thank you. I want to move on to the next speaker.